Welcome back, students. In this note set, we're going to take a look at the cell cycle with reference to a particular disease that I'll bet just about every one of us has been touched by, either ourselves or family members, cancer. And it's intimately tied up. The whole idea of cancer is a misfiring of the cell cycle. The relevant reading that you can find in your bio textbook would be in sections 8.8 to 8.10. Let's define something first. There are in our cells things called growth factors. So if we refer back to a diagram that we had taken a look at in our last note set, the diagram of a cell's normal life cycle, where there's G1 growth, a synthesis phase, and G2 growth, altogether comprising interphase, and then there's a period of cell division. During the course of that entire cycle, there are chemicals that signal when to enter a new phase. Given that most of a cell's life is spent not dividing, that is, in interphase, then when does a cell know that it's time to divide? Well, there's chemical signals known as growth factors. Now, students, you could spend years studying growth factors and exactly how they work. And in fact, many molecular biologists are doing just that right now in an effort to control cancer. We're trying to interrupt that disease. But in a normal cell, there are signals that come at a regular and proper time in a cell's life cycle, signaling when it's time to divide. Let's take a look at that system called the cell cycle control systems. And this includes all of the molecules, both the signaling molecules and the receptor molecules on a cell surface that trigger and coordinate key events. And if you can picture this as like a long journey that a cell undergoes, there are various stop points or checkpoints, you could say, for a cell to go through in order to complete its cycle. In fact, we know that there are 3 such checkpoints. We call one of them that occurs in the G1 growth phase the G1 checkpoint. Next, there's S phase, that's synthesis of DNA, a G2 growth phase, and then at the end of the G2 growth phase, just before mitosis is set to occur, there is something called the G2 checkpoint, through which a cell must pass, or it must have the growth factors that indicate that it's time to pass in order to enter mitosis. And then there's prophase, metaphase, and anaphase, and then before completing mitosis, there's a mitosis checkpoint, sometimes called the M checkpoint. So let's define those first, and then we'll take a look at some specific examples. The G1 checkpoint allows entry into the synthesis or S phase from the G1 phase in a cell's life cycle. It can also be the checkpoint that causes a cell to leave the cycle, going to a dormant or non-dividing phase called G0. So some of our cells are designed to go dormant for a period of time before re-entering G1 growth. The G2 checkpoint completes the second growth phase. So S phase would have been completed, so new chromosomes synthesized, and another growth phase completed. This is the checkpoint that allows the cell to go into mitosis, to begin mitosis. And then thirdly, the mitosis or M checkpoint is what allows the cell to enter anaphase of mitosis. So once it's through the M checkpoint, this cell is going to complete its cell division cycle and produce new daughter cells. Now, this is important. The default signal, that is the signal in every cell cycle control system, is to stop. That is to not let the cell proceed. So default means what happens if nothing else goes on. and every cell cycle control system is set to stop. The go signal is only given when the proper growth factors arrive at the surface of the membrane. So think back to plasma membranes and cell construction. There are proteins that span the plasma membrane, some of which are receptor proteins. The growth factor that triggers a response within the nucleus is one of those receptor proteins. So, or it binds to one of those receptor proteins. So usually a growth factor is a hormone, but it can be other things. Hormones circulate in the bloodstream, so they're broadcast widely throughout the body, but only a specific receptor is going to be able to bind it. Once the receptor protein receives that signal, there's a series of signal transmitting, we call it signal transduction pathway. When you get to college biology, you'll be able to study that in some more detail. But the idea is, upon receiving that somewhere in the nucleus, where the control system is being regulated and the nucleus is deciding when to go into different phases, then it allows the cell to go through one of the checkpoints. Our ability to understand the cell cycle control system may indeed be what enables us to combat the disease of cancer. So it's one of the most active areas of research today. Thousands and thousands of people are dedicating their careers to better understanding the cell cycle control cycle. By coming to a greater understanding of this, we might be able to disrupt diseases such as cancer. So let's talk a little bit about cancer itself, the disease. First of all, 1 in 5 people will die from cancer. So on average, 1/5 of the United States population dies from cancer in some form. Many, many more of us will contract the disease, but then either defeat it or die from something else besides the cancer. The reason it's connected to the cell cycle is cancer cells escape the cell cycle control system. They're not even regulated by the growth factors. And the result is the cells that are cancerous divide excessively. There's no stopping them from going from one round of division to the next. Instead of spending most of their time in interphase as a normal cell would, cancer cells are dividing constantly. And in doing that, they're invading other tissues of the body. And usually the result is you disrupt the function of some of those tissues, which can, lead to very bad consequences. Okay, take a look at this picture. Normal cells are dividing at a normal rate. Cancer cells are dividing at a much higher rate than normal cells. Cancer cells start out as normal body cells. They undergo some kind of mutation. We know about mutations from earlier in this unit. What exactly causes the mutation is also an area of active research. Exposure to high energy radiation may indeed increase the rate of mutation. But also, it should be noted, mutation is normal. A normal, healthy person undergoes mutation several times during their lifespan. It's not nearly as uncommon as we might think. But at some point, if a cell is to become cancerous, it will have undergone a mutation, and that mutation causes that cell to lose its ability to control the timing of its division. So it's not waiting for growth factors to go through the G1 and G2 and M checkpoints. It's just dividing constantly. And the result is disease is caused. Many cancer therapies are seeking to disrupt some of those steps in cell division. So can we target our therapies to prophase, metaphase, anaphase, telophase, or can we target our therapies to making a stronger G1 checkpoint, G2 checkpoint, or M checkpoint? If we can disrupt any of that, we could stop the cancer. But we have to do it in such a way as to not also inhibit normal cell division. So that's the tricky part. Okay, tumor. Tumor is a mass of abnormally growing cells within otherwise normal tissue. And there's two kinds. Benign tumors remain at the original site. They may disrupt the functioning of an organ if they just get bigger, but as long as they're not spreading, we still call them benign tumors. Benign doesn't mean nothing to worry about. It's usually something you would monitor and maybe undergo surgery to remove, but they're not the kind that are spreading. The kind that are spreading are called malignant. These are tumors that are spreading to other locations in the body. The process by which they do that is called metastasis. And that's the much more serious form of cancer. Or I should say, that is cancer. So just having a tumor doesn't necessarily mean you have cancer. It's having a malignant tumor that is when you have cancer. So let's take a look. Here's a breast tissue. Breast is a common source tissue in which cancer evolves in women, also men, but mainly women. So let's say there's a tumor. Initially, it's benign in breast tissue. As it gets bigger and bigger, it's invading the breast. And if some of the cancerous cells were to enter either the bloodstream or lymph vessels, then they're going to get carried to other parts of the body and they may take root there, and that's when it spreads. And if that happens, it becomes very difficult to treat and control. So what can we do? Ideally, we identify the tumor before it starts to spread. If it's a localized tumor, we can cut it out. You can go into a person's body, cut out the tumor, and maybe a minimal amount of tissue surrounding it, and that might do the trick. You can also treat it with concentrated beams of high energy radiation. There's proton therapy out at Fermilab in Batavia. In addition to doing physics research, they also do medical treatments. They shoot a proton beam at a specific location in a person's body, and in doing that, can destroy certain tissue, and you want to make sure you aim it at the tumor and not some tissue next to it. If the tumor has already become malignant and is spreading, it's through metastasis, a common treatment is chemotherapy. That means kill all dividing cells. It's a very painful, it's a very whole body type of process, and it's painful. So if you can treat the cancer without chemotherapy, that's definitely ideal. There are a lot of areas of hope in cancer treatment. Since we are able now to sequence the genome of just about any patient who comes to the hospital, it's becoming easier and easier to do this. If we were to take a sample of the cancerous tissue, sequence it with the help of a computer, and also get a sample of the DNA of a normal tissue in that person, we can figure out, of the 10 different treatment options, which one is most likely to help this person. And in doing that, not only are you not wasting time going through the other treatments that would be dead ends for that patient, but you're saving them a lot of expense and time and effectiveness, just tailoring. So the idea is personalized medicine. We're hoping to get more and more data to target a treatment to a specific person. And if one person has a certain type of cancer next to another person with that same type of cancer, their treatments might look very different based on their genetic profiles. And again, this is an area of active research in biology. Students, I hope some of you who may be motivated by this and want to help, please learn as much biology as you can. This sophomore level course is just an introduction to biology. There's so much more to learn. And I hope you carry this forward through elective courses later in high school, some biology courses in college, maybe some grad school. And then you could be working in a lab before you know it, really contributing to this major problem.